So what happens when the insect is able to break through and encounters potential toxins that the plant has, but the toxins haven't quite made it into the body yet? So what are ways in which insects can actually deal with this? And we're just going to talk about one really common one, and that is that insects have all kinds of tricks to actually deactivate or to make less toxic the compounds in the plant straight as they're taking it in. So here's a well-studied example. And one is through the production of enzymes in the saliva. So as they're feeding on the plant, they're secreting salivary juices over it. And in the saliva itself are a series of proteins that actually stop the plant cells from actually producing some of those compounds. One of them is a well-studied enzyme called glucose oxidase that seems to interfere with the signaling that the plants use to say, oh, it's time to produce a secondary compound that may have the capacity to deter or fend off an herbivore attack. So this is one way in which insects can actually interfere with this. And then there's other ways that they can also do it through the production of plant growth regulators as well. But I'm going to talk mainly about the first one. So here's a really neat study that a group did where they actually disabled the spinnerets. This is the place where the saliva actually gets secreted by certain caterpillars as they're feeding on the plant. And what they found is that if you look at caterpillars that don't have their salivary glands intact, the ability of the plant to produce, these are feeding on nicotine, nicotiana, so nicotine-producing plant, and nicotine is one of those alkaloids that is inducible and actually can be quite toxic to generalists. When they didn't have their salivary glands intact, there was a lot of nicotine, this neurotoxin being produced. On the other hand, and this is without the glands, when the glands were left intact, and you can see here the kind of surgery that they did with these caterpillars, you kind of cut a little slice through them without being too damaging. You can pull the salivary glands out, you can clip them with a small pair of scissors, and then you can kind of suture them up like this. You can kind of cauterize them shut. This is how you remove the glands, by the way. On the other hand, when the caterpillars have the glands intact, you find a big decrease in the production of the nicotine. And by the way, this is what it looks like. This is how much nicotine is in the plants by themselves without any herbivores on them. So the activity of an herbivore induces the production of these neurotoxins for the insect, but the presence of the salivary glands in the caterpillar can actually decrease that production. So there's something in the salivary glands that actually prevents the plant from making as much of that compound as they otherwise normally would. And one of the key elements of that is this enzyme called glucose oxidase. And you can do certain experiments like induce the plant, poke the plant in various ways, such as by just poking it with water and then seeing how much, making a wound and putting water in it and then seeing how much nicotine is produced. Taking a wound, which is a lot, wounding a plant with a little forceps and then inserting glucose oxidase that's been heated and therefore deactivated. Remember, these are proteins and they're sensitive to denaturing in a variety of different ways. On the other hand, when the glucose oxidase is still functional, that hasn't been denatured, then the production of that nicotine is reduced. And so this is one way in which people have figured out that it's not just saliva in general, but it's a particular component in the saliva, this glucose oxidase enzyme that's produced by the insects, that can suppress the production of these plant secondary compounds. And of course, you can take saliva and you can add take saliva with active glucose oxidase. This is kind of the normal bug spit that's coming out. And you can see what actually happens to these, which is about the same as just having the glucose oxidase. And interestingly here, the amount of nicotine production when that's not wounded at all is about the same as with the saliva actually acting. So they've pretty much shut it down shut down the induction process completely.